Welcome to WinCafeSci Online. A recording of this talk will be posted on our YouTube channel, and you can find details of this and previous talks, as well as our forward program, on our website, which is www.wincafesci.org.uk. If you enjoyed tonight's talk and you're not on our mailing list, you can sign up for it at the website to be notified about future talks. Tonight's speaker is a postdoctoral researcher in the Wolfson Centre for Mathematical Biology, part of the University of Oxford's Mathematical Institute. His research focuses on developing mathematical and statistical tools to describe immune cell interactions with cancer, and on integrating these techniques with medical imaging software to provide practical tools for medical researchers and pathologists. His work sits at the intersection of mathematics, computer vision, statistics, immunology, and oncology. Other awards include winning the official Premier League fantasy football competition. Who says maths is just for nerds? Please welcome Dr. Josh Bull.
Thank you, and thank you very much for asking me to speak. So thank you very much for that introduction. So I'll just start off by, if I can change slide. Fantastic. Start off by telling you a little bit about myself, about where I work. So this is the Mathematical Institute in Oxford, which is normally where I work when there's not a pandemic on. So at the minute, I'm just working from my front room, and one day I'll get to go back to this nice building and do some maths there. So I'm a postdoctoral researcher there, and I did my PhD there as well. And I'm in the Wolfson Centre for Mathematical Biology. So my research specifically looks at cancer and the immune system. And in particular, I'm interested in spatial interactions between these. So we'll go into a little bit more detail of what that means in a bit, but essentially looking at how these things are physically interacting in a kind of complex environment of a tumor. So I would kind of summarize myself as sitting in the middle of lots of different disciplines. So in particular, I use a lot of tools from image analysis. So this is some code that I used in my PhD to identify cell locations within a tumor. But also a field called spatial statistics, which tries to describe kind of patterns that we see in spatial data. So the blue dots that you can see up here are the locations of immune cells within a tumor. And we'll think a bit more about later on about how you can describe that. And I also do a lot of mathematical modeling, which will be the main focus of this talk, of what a mathematical model is and how we use them. So the field of mathematical biology is, for a mathematical field, it's very young. So it's only actually been around since the 1950s. And it was kind of kick-started by Benedict Cumberbatch when he invented the computer, or more accurately, Alan Turing, who's obviously better known for his work doing code breaking in World War II and as the face of the £50 note. So after leaving Bletchley Park and going to the University of Manchester, he kick-started the field of mathematical biology. And my own pet theory for this is that biological systems are a very complex thing. There's lots of interacting parts. And that's something which mathematically we struggle to describe. So we're familiar with the idea of using equations in something like physics, maybe in chemistry. But the idea of using equations in biology is much harder because these systems are just a lot messier. And my pet theory is that if you'd happen to have just revolutionized the computer, that might be a field which really applies to you. But I should stress that I have no evidence for that claim whatsoever. It's just my pet theory as to why that was the problem that he turned to. But in particular, his first work on this was called the chemical basis of morphogenesis, which introduces a set of equations. So you don't need to understand these equations, but essentially it's a set of 3 interacting chemicals. And the idea that he was putting forward in this paper was that if these chemicals diffuse through a tissue at different rates, they might interact in ways to form interesting patterns. So in particular, this is one of the images from that paper where he talks about making these kind of dappled patterns. So I think this one is supposed to look like the side of a cow. And this was really interesting because obviously these kind of patterns are something that we're really familiar with in nature. So if you look at a tiger or a zebra or any animal that has stripes or spots, there was always a bit of a question of where that came from. And Turing showed that you could get these very complicated patterns arising just from a very simple set of 3 equations, or you can even do it with 2 equations. So a good example of this is some work that came out of the Center for Mathematical Biology, where what you see on the left are some real fish, and on the right is a simulation solving 2 or 3 of these equations to produce similar patterns. And in particular, you see these interesting kind of artifacts where you get breaks in these rings, which are things that you see in real animals. And similarly with the stripes on these fish, the types of pattern that emerge from these very simple systems can be very complicated. So these types of patterns are known as Turing patterns. And actually they now appear on the New England football kit. So we were kind of amazed to see this a couple of weeks ago, where I'm sure that this pattern here has been made by a mathematician, but I'm yet to find out who's made it. But these ones were made by Andrew Krauss, who's a researcher in our group, just to prove that it looked very similar, basically. So Turing patterns were where mathematical biology really started. But one of the great things about maths is you can apply it to lots and lots of different problems. And in particular, biology is a very broad field. So one of the most relevant places that we find mathematical biology at the moment is looking at epidemiology. So in particular, when we're doing forecasting for the coronavirus, when people are saying, this is what we think will happen, that tends to be based on these mathematical models. So mathematical modeling has kind of hit the news over the last 6 months because of that. But actually, the basic techniques for doing that, again, as with Turing, are very simple systems. So in this particular model, this is known as the SIR model, and it just describes the rate at which susceptible people become infected and at which infected people recover. And using just those very simple interactions between groups, you can get very complex dynamics. Another place where mathematical biology was really first used is in describing predator-prey relationships. So if you're a vicious predator like this fox or the prey that's underneath, you tend to see these interesting dynamics where having a large amount of prey animals leads to an increase in the number of foxes, the number of predators, but then the foxes are killing more of the prey and the population can't sustain it, and you end up seeing a population collapse. Those types of dynamics, again, are really nicely described by just a couple of equations. I also wanted to highlight some more medical uses. So networks at the moment are a very hot area of study. So they've been studied in mathematics for centuries, and now they're becoming more and more relevant to looking at the way that we design our transport systems or the way that human brains work. And then maybe from a more traditionally mathematical point of view, fluid mechanics is a very difficult problem to solve. And mathematics does a very good job of describing it. So all of these problems kind of come under the area of mathematical biology. So it's a very, very broad field. And there'll be researchers who work in multiple ones of these areas. So some of the techniques that you can use to study epidemics, you can also use to study predator-prey systems, for instance. So we tend to either be a mathematician who's got a nice technique that they like and will apply it to lots of different areas of biology, or you might be somebody who lives more focusing on one specific problem and uses tools from all across mathematics to try and deal with that problem. So I'm probably in that last category, and I look specifically at the maths of cancer. So this is an immune cell attacking a tumor cell, or at least an artist's visualization from the BBC. So what is cancer? Well, again, that's a very complicated question to ask. So at the highest level, cancer is essentially uncontrolled cell division, uncontrolled growth. So in this particular case, one of these normal cells, while most of the normal cells will reach a point where they say, oh, I need to stop dividing now, we've filled up all of the space that we need to fill, and now I'll stop. One of those cells picks up a series of mutations, and it enables it to just keep growing and growing and growing. And fundamentally, that's what happens in all types of cancer. It's just a single cell, possibly, that's gone out of control and grown into one of these growths. So that's the very high level explanation. But actually, if you look in a bit more detail, there's a lot more to it than that. So the reasons that tumor cells continue proliferating there's a lot of reasons, right? So it's not as simple as a cell just gets a mutation. It has to do things like avoid the immune system. It has to be able to spread into different tissues. It has to be able to outcompete the rest of the cells around it. So even once you start looking at the question in a little bit more detail, it becomes much more complex. If you dive into the real deep biology, you end up with something which looks like this. So this describes all of the chemical interactions going on, just one particular signaling network. So when one particular tumor cell receives a signal through whatever this mTOR network is, all of these interactions are happening. And there are literally thousands of these, and we don't know all about them in any kind of detail at all. There's a lot of great research in biology and research in the clinic, which is trying to uncover more and more of these interactions. So when you start looking at it from that point of view, it starts to become a bit clearer why we have so much trouble being able to cure cancer, because it may be any, this is one tiny part of the network of what's causing a cell to divide, say, and there may be a problem at any point in this. And a treatment which works for one tumor may not work for another tumor because the mutation is slightly different. So this, for me, is where mathematical models come in, because our aim is to try and simplify biological systems so that we can understand them better. So there's a really nice metaphor for this that I like to do with maps. So you can ask the question, what is the best map of London? So strictly speaking, the most accurate map of London would have every hill, every tree, every blade of grass, every pothole. All of that information is something that you could have on a map. But obviously that would make the map the same size as the city. And although it would be very, very detailed, it wouldn't really help you to find your way around. So something that we do is we simplify away some of the complexity. We don't need to know where every blade of grass is in Regent's Park. We can just say it's this big green blob. And that gives us a good map. We can use this map if we want to know how to get from the British Museum to the Imperial War Museum. The map that you can see at the moment is great for that. But it's not necessarily the best map of London. So if I was to say, well, I'm not interested in where the British Museum is, but I need to get from Liverpool Street to Paddington, then this is probably going to be a more useful map. And if you were comparing this with the satellite image on the left, you would say, well, that's not a good map at all. All of the train stations are in the wrong places. The distances are completely not to scale. We've got all kinds of problems with it. But just because it's wrong doesn't make this map useless. It's still a valuable map. And in fact, if it's telling you, if what you want to know is how to navigate the tube system, this is the exact map that you need. You don't really care about all the rest of that complexity. But then equally, if I wanted to know where I could cycle within London, I need a different map again. So all of these maps are telling us the same information, but in very different ways and focusing on very different aspects of that information. So there's a very famous quote in statistics, which it's obligatory to say in every talk about mathematical biology, which is that all models are wrong, but some models are useful. So strictly speaking, none of these maps is an accurate description of London, but all of them are useful in different ways. And that's what we try to get across when we're making mathematical models. So our mathematical models aren't trying to be a perfect description of the real world. We're not trying to involve every single interaction in that diagram that you just saw. We're just trying to get a feel for how the world is working at a kind of higher level, which means that we want to skip over lots of irrelevant details so that we can focus in on the details that are important. So in particular, when we make a model, we want to design it for a specific purpose. We don't want a map of London generically. We want to make a map for cycling, or we want to make a map for navigating the London Underground. It's exactly the same with mathematical models. We don't want to design a model just to try and capture all of cancer. We want to try and answer a specific question. So a lot of the art that's in mathematical modeling is in knowing what the right question is to ask, and then being able to build a model to address that particular question. So thinking about the highest level description of cancer, which I introduced a moment ago, well, the highest level model that you might ask is to say, well, how many tumor cells will I have? How many cancerous cells will I have after a certain amount of time? So this follows the dynamics of exponential growth, which is what happens when you have one cell, which becomes 2 cells, which becomes 4, becomes 8, becomes 16, and so on. So the simplest cancer model that you can get really is something exactly like this, just some e to the power of some number. And this will tell you after a certain amount of time, how many cells do I have? And actually that works very well at answering that question. If you put a tumor cell in a dish or something and give it plenty of oxygen, plenty of nutrients, everything that those cells need to grow, they'll keep growing following this exponential growth curve. But actually, knowing that doesn't necessarily help us to say anything useful about how tumors act within a person, which doesn't get us any closer to being able to help find some kind of cure. Because what happens is in a person, when we get cancer, these tumors are being supplied by oxygen that's coming from the rest of the body, coming from blood vessels. And those blood vessels can only support a certain number of cells. So as you get more and more cells piling up, there's not enough oxygen to go around. And so actually you don't see this kind of dynamic, you see some more complicated things. So a model, different kind of model, an experiment, perhaps is a better way of putting it, which represents this in a lab, is something called a tumor spheroid. So this is what an actual tumor spheroid might look like. And essentially what you do is you put some tumor cells into a dish, a test tube, and you let them keep growing. And what happens is they form into a kind of ball like this, but they're getting their oxygen from the fluid that you've put around them from this kind of growth medium. And so you get some dynamics that happen here, where we have oxygen that's coming in from the edge of the spheroid, and the cells are all taking it up. But the oxygen is diffusing in towards the center. So the cells at the edge of the spheroid are very happy. They've got lots of oxygen. They can proliferate away as much as they want. But the cells that are pushed into the middle are getting less and less oxygen. And eventually they reach a point where they can't survive anymore. So we call these cells that are not getting enough oxygen hypoxic. And you end up with what's called a necrotic zone in the middle, where the cells can't survive. So this is something that you see in experiments. So these are cross sections of tumor spheroids. And in these top images, the green cells are happily proliferating and the red cells are dying. So you get this very interesting structure. And this is something that we do see within people as well, because the cells which are close to the edge of a tumor, or that are close to blood vessels, will be able to proliferate a lot more than the ones which are far away. So this is often used as a kind of, I am going to say a model. It's a model system, but not a mathematical model. It's a biological model. But we can make a mathematical model of that biological model and maybe capture some more interesting dynamics and make our model more descriptive of what tumors are like in real life. So I'm going to introduce you to this next model up. We've seen an exponential model. This is a more complex model called the Greenspan model. So this image has come from a paper that's about applying radiotherapy to tumors. Now, you're not interested in capturing all of these different bumps in the edge of a tumor spheroid when you're interested in describing the broad effect of treatment on that. You're more interested in describing these dynamics. So what the Greenspan model does is it splits our, instead of saying we're going to count the number of cells, we're going to look explicitly at space. We're going to look at where those cells are. So if our cells are in a very low radius, if they're close to 0, they might well be in a necrotic compartment where they're going to behave differently to cells which are near the edge of the spheroid and are able to proliferate. So what the original model looks like is something like this. So don't worry if you can't understand the maths. Essentially, the aim is that oxygen is diffusing in, so it comes into the spheroid from the edge, and it's consumed by cells. And then we have another equation which describes how the tumor volume changes in terms of cell proliferation and cell death. So when we add all of those dynamics in, we don't just get this exponential curve anymore. We get something which looks more like this. So at small times, when we have a very small spheroid, the number of cells is growing very quickly. But as our spheroid gets larger and larger, the amount of proliferating cells at the edge balances out with the number of cells in the center that are dying. And you see spheroids reach this kind of steady state size where they're no longer getting any bigger. So this is a type of behavior that you see in people. But again, it's a simplification. We're not capturing all of the information. We've got more detail. We're including oxygen now, but there are a lot of other aspects of cancer that we're interested in modeling. So I've mentioned a couple of times that my main research interest is immune cells. So we might ask the question, well, where do they fit into this? You know, how do my immune cells get into this tumor? So it's helpful to think of a simpler question first, which is, well, I could add some immune cells onto this tumor and see where they go, but there are a lot of different things that they could be doing. They might be being pushed around by the fluid that's around my tumor, or they might be responding to different cues that are coming from within the tumor. So there's a set of experiments that I'm going to talk about a bit, which are a simplification of that. So rather than saying, what do immune cells do in a tumor? What these experimenters did was they just took some plastic beads and put them onto one of these tumor spheroids and said, well, what happens? And the really interesting thing is, even though these are just plastic beads, you see these beads start to move towards the center of the spheroid. So this is some data that's taken from that paper. So on the left here, we're looking at the spheroid edge. So at early times, all of the plastic beads are just found at the edge of the spheroid. But even though they're completely inert, as time goes on, they move in towards the center. So this is a really interesting phenomenon. It's a simplification of looking at immune cells. We're taking away a lot of the complicated things that those cells might be doing and saying, let's just see what effect the tumor has on some particles. So in particular, what we're seeing here is a pressure gradient. We've said already that cells at the edge of the spheroid are proliferating and cells at the center are dying. So what happens there is all of this growth at the edge is pushing cells inwards, and that forms a kind of conveyor belt that's able to carry this plastic in. So there's an interesting question there, which is, well, what impact does that conveyor belt have on the immune system? If we're interested in immune cells attacking tumors in real life, is that conveyor belt going to help us because it's carrying them in? Is it going to cause problems for us? We don't know without doing some experiments or without doing some modeling. So one approach that you can do, if you're my PhD supervisor and the year is 1999, is to make a model of this. So this is an adaptation of the type of model that you just saw. We're only interested in radius. We're forgetting about the rest of the complicated space, and we're just saying, how close are we to the center of the tumor? And they do see in this model an approximation of what we've just said. They see this is the distribution of beads, and we see them get carried inwards towards the spheroid center. But you might say, well, this is too much of an oversimplification. I'm interested in these experiments where we literally took some blobs, some beads, and we put them into this spheroid. And if I go back to that picture that we saw, well, some of these are closer to the center than others. They're not nicely distributed at the same radius. So this is a bit of an oversimplification that we've got. So we need a more detailed map. Our model is helping us, but it's not quite able to capture our intuition of what we're interested in. We don't have these nice discrete points, and we're not seeing them moving around. So one class of models that's particularly interesting are known as agent-based models. So in agent-based modeling, you simulate every cell as its own agent. So each cell has got the ability to make decisions for itself. And you can make this as complicated as you want. So each cell might have its own cell cycle model that tells it when it's able to divide. It might be able to respond to oxygen or not. What we might do is calculate the forces that are acting between all of these cells and end up with some sort of simulation like the one you can see here, which is exactly what I was talking about. It's a tumor spheroid simulation. And you can see that the cells in this model are dying in the center once the spheroid becomes large enough. So there are quite a few ways that you can do this. So the one that you're seeing here is from Paul Macklin's group in Indiana, using a bit of software is written called PhysiCell. In Oxford, we use this piece Chased quite a lot because that was developed in our group. And that's the work that I'm going to show you now is some work that I've done within this Chased environment. So in particular, the next few slides are very much hot off the press. So this is a paper that we had published about 2 weeks ago in a journal called PLOS Computational Biology to look at this experimental problem from the 80s and from the 90s, and to really investigate it using today's techniques and see if we can learn anything new. So we're going to model every cell in a tumor spheroid as its own separate agent. They have their own rules, and we're going to update their position based on the forces that are acting on them. And they're going to move between being proliferative at the spheroid edge to being hypoxic in the center and ultimately dying. So my graphics don't look quite as nice as Paul Macklin's, unfortunately, but the model underlying it is the same. We have the spheroid, which is growing, and once it reaches a certain size, we can see that the growth levels off. It kind of reaches here and it's not able to push anywhere beyond that. So reset. So if I plot the growth curve, it looks a lot like the one that we saw in that Greenspan model earlier. It grows quickly at the start and then it tails off once it reaches a set size. But now we can add our microbeads to the edge, and we can see what happens as they move inside the spheroid. So over time, they get internalized, and we can do exactly what they did in the experiments and show that if we set our model up correctly, we get the same result. Right, brilliant, great. what have we learned? Well, not a lot. We did some experiments back in the 1980s, and the conclusion that they reached was this is because microbeads are pushed towards the center. And our model says exactly the same thing. So you might say that we've put in a lot of work to not get anywhere. But this is where the real power of mathematical modeling comes. Because within the experiments, experimentalists are very limited by what they're able to do. They might have, so one restriction with those experiments, for instance, was that they only had a certain number of Tumour steroids, and they had to essentially cut them open to see where the beads were, which meant that they could only do it every now and again and with a different spheroid. Whereas what we're able to do in a model is track the individual trajectory of each one of these beads. So this graph here, at the top of this graph, we've got the edge of the spheroid, and at the bottom, we've got the spheroid center. And this mark at the time 300 is where we add those beads to the edge of the simulation. And then all that I'm going to do is over time, I'm just going to plot where they go. And we can start to see two different behaviors going on. At the edge of the spheroid, we've got lots of random bouncing around. Microbeads just being kind of pushed this way and that. But once they kind of cross this line, or once they get towards the center, they move kind of just inwards. So we've almost got two different regimes. We've got one where they're being bounced around and one where they're sitting on this conveyor belt. And effectively, what we showed that you could do using a mathematical model is to measure the speed of that conveyor belt. So if you can do modern experimental techniques which let you trace the location of a bead over time, we showed that you can actually use that to measure the speed of this conveyor belt. And more importantly, you can use that to make predictions about what's going on in the model. So the main reason that this figure is here is to impress you all with how close all of these dots are to the line y equals x. So on the bottom here, we've got a parameter that we use in our model. So we set our model up and we say, well, the growth rate of cells is going to be, they're going to divide every 20 hours. And then we say, well, can we predict that? If we then run that model, run that simulation, and track those bead trajectories, can we measure this and from this graph predict what that value was that we used? And the answer is yes, we can. So what we've got now is a way of going from things which are measurable experimentally to parameters in our model which describe how the tumor cells are growing and evolving and responding to different inputs. So then the following question is then, well, okay, can we go and revisit those experiments? Can we take the knowledge that we've got from this model, that if we measure these things, we can talk about what the division time is for this cell line and how the cell line will respond to hypoxia? And can we go back to those and predict what those things are just by measuring these bead trajectories? And the answer to that is, I don't know, because we haven't yet done those experiments. But this really is representative of the process that we're trying to do in mathematical modeling. We want to take a biological question and a phenomenon which is seen in experiments, but perhaps isn't fully understood, and then use a mathematical model to really investigate that and see what we can change in an environment that we can control, to then be able to go back and revisit the experiments with a new hypothesis. So we started off by asking about immune cells, and I immediately started talking about plastic beads instead. So you might say, well, Josh, what about those immune cells? Do they get carried along by this tide, like James Blunt at whatever concert this was? Are they just pushed by the beads, by the cells that are around them? Or do they have some way of pushing through? Can they force their way into a tumor? Or indeed, are they resisting this flow? So the question then becomes, well, what impact does this conveyor belt have on immune cells in tumors? And that's something that you can ask quite happily with a mathematical model, because with a model, I can start 3 simulations from exactly the same place. So I know what I don't, in an experiment, there will always be differences between the ways that I do things. Whereas I know that each one of these 3 simulations that you're about to see has got exactly the same amount of oxygen available, exactly the same proliferation rate, the same response to hypoxia. So I can control for all of those things. But I can add in my model, for instance, attraction to hypoxia. So this is a mechanism that we see in real life, where immune cells sense that there are hypoxic cells, cells that don't have enough oxygen, and they move towards them, because that's often a sign of a wound. So if you're, say you get a small cut, well, one of the first things that happens is you want immune cells to go there, because there are going to be bacteria and things that are getting into your body through that cut. But what that cut does is it disrupts the oxygen supply. It's breaking those blood vessels. And so the cells which are there, which are hypoxic, start releasing all sorts of distress signals that attract immune cells. So we can simulate those distress signals in this model, which is exactly what you're about to see. And we can say, well, I can control how sensitive my simulated immune cells are to that signal. So in the left-hand case, we'll say, well, they're not really interested in it. They're just going about their own business. But as we move towards the right, they'll be much more sensitive to that and much more attracted to those hypoxic cells. So if I'd set all of those simulations going, when we get to 300 hours, I'm going to add in these immune cells at the edge. And we can see that they start to move into the spheroid. So in this left-hand one, where they're not really interested, we have a very few in the center and lots kind of sat around the edge. They're not really interested in invading the spheroid particularly quickly. And what we can do is we can compare this against the bead simulations and say, well, are we seeing more or less infiltration than we would have been if these were just plastic beads? And obviously, as we increase their attraction, we see more and more microbeads that are invading those spheroids. So if I just put those on again, if you watch the right-hand side and compare it to the left-hand side, see how quickly they move in comparison. So the important take home from this is that in a model, we can make sure that we're only changing the thing that we're interested in. But we can then take this and we can compare it with real data. And we can say, well, we can make measurements of a real tumor. And we can use that to measure whether immune cells are responding to this or not. So the equivalent experiment for this might be, what if I could grow a tumor spheroid and engineer it somehow magically to only produce one certain distress factor? And I could then see, are my immune cells doing this or are they doing this? And if I know that, then that will tell me something about how immune cells are responding to that particular chemical when they're in your body and they're surrounded by all sorts of conflicting signals that are going on. So in real life, these spheroids are again, an oversimplification. So we're building up complexity. So this is an image from a tumor. And the brown blobs that you can see in here are immune cells. And the blue is tumor, essentially. And what we can do is do some image analysis to pull out the locations of those cells. And we can also look at adjacent regions of this tumor to see where the tumor cells are. So these brown ones here are tumor cells. And we can see that we've got some healthy tissue here. The tumor is kind of surrounding. And we can even look at the amount of oxygen. So these brown cells are hypoxic, and you can see this region in the middle where there aren't really any cells is our kind of necrotic zone. So we have broadly the same structure as within a tumor spheroid, but the geometry is very different. We no longer have a nice blob, a nice sphere. We've got a very complicated surface where at the edge here, these are the cells which have enough oxygen. And then there's a very complicated boundary before you reach the cells which are dying at the middle. But we can hypothesize, well, if we've got this region of death and region of proliferation, do we have the same conveyor belt effect going on? And experimentally, this is very hard to determine. But with a mathematical model, you can take the exact same model and apply it in a new context. So in the simulation that you're seeing now, we've taken a real piece of geometry and taken the information about where the tumor is, where the oxygen is, and where the immune cells are. And we can run our same simulation in this more complex geometry and start to see these individual immune cells moving around inside the tissue. So that type of predictive modeling is really interesting, because now we have images which are relatively straightforward to get hold of experimentally, which show us where immune cells are inside a particular tumor. And we have a model which tells us where immune cells are inside a particular tumor. So we can use this in these more complex geometries to try and compare between the two. But that then leads to a question, well, how do you do that? How can you describe, if I said to you, where are all of the green dots in this image? It's not an easy thing to describe because it's a very qualitative judgment. Oh, there's like most of them are over there. That's not something which is very mathematically rigorous. So one of the other areas that I'm interested in, aside from mathematical modeling, is in how we answer that question. How do we describe where these points are, where immune cells are within an image? But more generally, if I've just got a load of points, how do I describe what those look like? How do I make that qualitative judgment into something quantitative? So we see this in lots of different data sets. And in our simulated data sets, we can control the amount of complexity. So these simulated point clouds are essentially just me saying, I'm going to put some dots down, and I can control exactly where those dots go. At a slightly more complex level, I could think about where my beads are within an agent-based model. But then I might say, well, what if I take a particular sample of a piece of tissue? We've got a little bit more complexity. Where are the immune cells going within that? Or if I look at a region like the one that we've just seen, where are they there? And ultimately, I want to know, if I've got this whole complex tumor geometry, where are my cells there? How do I describe immune cells in this tumor when they may be in very different places, in very different areas? So one of the ways of doing that is to think, well, what is it that we see when we look at an image like this? So these points are the locations of immune cells within a tumor, but now we've just got those X and Y coordinates. And the types of things that we tend to be interested in as a person are things like whether these points are clustered together. Are we seeing particular dense groups where these things are all sticking there? And in particular, are we seeing any areas where we don't have these dense groups, where we have maybe a point that's off by itself that may have infiltrated into our tumor? And those things are quite difficult to describe. The main reason for that is that traditionally the way that this has been done by clinicians and by pathologists is by measuring things like immune cell density and doing some very subjective scoring about that. But the trouble is, even measurements like the mean or the density don't necessarily tell you about the shape of a point pattern. So this is my favorite data set in the world, which is known as the Datasaurus dozen. So there's the Datasaurus himself. So every frame of the video that you're looking at has the exact same number of points, so the same density, but all of these images have the same mean and standard deviation in both the X coordinate and the Y coordinate. They have the same correlation coefficient. So the types of measures that people normally use to describe point data are giving you exactly the same measures for all of these things. But qualitatively, all of these images tell you different things. So part of the moral of this story is we'll always look at your data, because there may be some underlying pattern there. There might be a dinosaur hiding in it. But part of the moral of the story is, well, maybe we need some new mathematical techniques to be able to describe these patterns. So some of the areas that I work in are things like spatial statistics and using topology to analyze data, which are quite in-depth techniques, which are talking themselves, I guess, but essentially both try to answer the question of how do we make qualitative descriptions something that we can quantify. So thinking about the image we saw a moment ago, we may be interested in whether there are clusters, or whether there are large gaps and voids in that image, or maybe whether the cells are close together or not. Those are all qualitative descriptions, but those are also things which we might be able to describe quantitatively. So the four regions that you're seeing here, on the left we have a section of a tumor image, and on the right is the X and Y coordinates of the immune cells. So we asked a pathologist for each of these images to rate the amount of infiltration, and this very low, low, moderate, high is what they said. So essentially this first image is representative of immune cells which aren't going into a tumor. They're kind of sitting around the edges of this shape. Whereas as we move further down here, we get immune cells and tumor cells are kind of fairly well mixed. So you may look at this and say, well, this top image has lots of empty space. It has lots of very dense clusters. That's how I'm going to make my judgment. But actually, we can do some statistics. And I'm not going to tell you what these are, except that this peak up here is telling you whether there's a cluster or not. This star down the bottom here is telling you how large the gaps are. So in my purple image, I have very large gaps between cells, whereas in this yellow image down here, I have very small gaps. And finally, we have the distances between cells. And you can see the types of pattern which arise when you compare these things. So without going into any detail of what these techniques do, you can use these to quantify some of our intuitions about what a pattern looks like. And ultimately, you can then use these to compare one of these images directly with the output of one of our agent-based models. So the take-home message from this really is that research is an ongoing cycle. It's something where we're constantly trying to simplify what we're modeling, but then at the same time, we're constantly trying to build on our models to make them more complex. So this diagram, I think, describes this really well. We want to go and we want to use experimental data and use our collaborations with clinicians and with doctors to try and come up with biological questions, try and find those areas of tumor biology that we don't understand. And then using our model, we can make predictions that can then be tested experimentally. We want to move this process round and around and around. And really, mathematics can help with the modeling, but it can also help with the comparisons, as we've just seen. We can simulate immune cells going into a tumor, but we can also use mathematical techniques to make qualitative judgments much more numerical. So really what we're trying to do is use simple models to describe the world, to describe how the world works, and to try and build on our understanding of those models to ask deeper and deeper questions. So I think that's about 45 minutes. So that brings me to the end of the talk. Apart from the last figure, which is the effect of an impending thesis deadline on the amount of caffeine that you consume. I can confirm that as the deadline day approaches, the number of cups of tea grows pretty much exponentially. Thank you very much for listening. And yeah, I think there's some time for questions, I think.
Okay, well, I think we're ready to kick off with the question and answer sessions now. Before we do, I have to say I really will treasure that quote of yours, Josh, about all models are wrong, but some are useful.
It's a good one, that.
Good,
But
good
it was fascinating also to see the complexity of the cancer interactions and processes on that early slide. I mean, that was really mind-boggling that there were so many different things going on, compared with the common popular perception that cancer is cancer, and very clearly it's so very many different things. So what have we got? Caroline saying thank you, that was an excellent talk. Andrew Sieber, do you want to put your question yourself?
Can you? Am I unmuted?
You are.
Oh, good. Yeah. Okay. Well, first question was: are there links between this work and the mathematical basis of artificial intelligence? And are there sort of parallels to be drawn there?
So in terms of developing artificial intelligence, or of using neural networks in application?
Yeah, there seem to be similarities in the complexity of what you're trying to achieve. I wonder whether there are sort of cross-fertilizations between your work and theirs.
Yeah, so I think particularly from the more networky end of things, there's a huge amount of work that's going on trying to describe these kind of very complicated networks of interacting things. So there's a lot of work in computational neuroscience, which is a kind of subset of mathematical biology, if you like, where people are trying to break down brain networks and really try and understand what different parts of that are doing. So whether that research can then be applied to developing, say, an artificial brain or something like that, I'm not entirely sure, but I think I would expect to see the similar kinds of structure. And I guess that research did inspire kind of neural networks as a technique of what is machine learning effectively. And that the kind of structure of a neural network is very heavily inspired by neurons, by a brain structure. So there's definitely cross-pollination there.
Yeah. Okay, thanks very much.
Yeah. So yeah, Carol said most interesting talk on a very complex subject. Don, do you want to ask yours?
Yes, it was really a fairly simple question. I look at your mathematical models of something that's in the natural environment, and you're really looking at anomalies and complexities of that sort. But I wondered whether you got overridden in some way by the natural environment's tendency to build fractals. And you may find that some of your models are dictated by this external influence without you even having created the opportunity.
Yeah, so the real problem is when you think about complexity in a kind of technical mathematical sense, where with what chaos theory essentially says is that things are very sensitive to the environment that they're in. And so no matter how perfect our description of a particular piece of biology might be, that's happening in this very messy environment where we don't have perfect knowledge of all of those conditions. And so that will then cause kind of effects which may or may not kind of spiral. So our predictions may or may not get much worse. The nice thing about using a mathematical model to kind of really try and examine a particular biological question is that you can control for that. So you can really, when you're formulating your mathematical model, you can look at how all of the interacting parts are there. And essentially, what you're trying to do is remove as much complexity as possible from that model so that you've got the most basic possible description that still explains the behavior that you're seeing. So maybe a good analogy for that is the original model that I showed you briefly from the 1990s, which was describing these beads, is very simple. It doesn't look at each individual bead. It just says, we don't need to worry about that. We're just interested in the kind of bulk behavior. So that is able to answer questions like, is there this pressure gradient? But if you want to look at more complex questions, then you need a more complex model. So keeping the model complexity low is a good way of keeping a handle on whether you're going to get this unexpected behavior that creeps in or not.
Okay, thanks. Mike, do you want to unmute and ask your question?
Yeah, okay, hi. Yeah, thanks, great talk. Are you working with any specific clinicians or any specific materials that you're looking to see how they migrate?
Yeah, so in terms of working with clinicians, basically everything that we do as mathematical biologists is driven by the biology. And we're very much aware that that's not where our expertise is. So when I started my PhD, I hadn't touched a biology book since I was about 16, I think. So we're very aware that although we're able to read that literature and get some understanding, we're not the experts in that. So for every model that I make, essentially, we're working with a group of clinicians that are driving the kind of biological question that we're trying to answer. Does that answer your question?
Yeah, I was thinking, you know, whether you've got specific programs on specific cancers, or, I mean, are you working, you know, with one dedicated team, or are you working with several teams? I was trying to sort of understand a bit more about how that interaction works.
Yeah, so very much with several teams. So there's one group that we collaborate with who are experts on immune cells, and in particular immune cells in hypoxia. So in fact, it's the lab of Peter Ratcliffe, who won the Nobel Prize last year. So within that lab, and that was for work on hypoxia, basically. So we're trying to bring our techniques to their experiments and see whether we can use those to understand specifically questions around hypoxia for them. Whereas there are other groups that I work with that are interested in completely different biological kind of effects. So still looking at immune cells, but maybe looking at how those cells are recruited or how those cells move within a tumor. So we're then working with different biologists on different aspects of whatever it is that they need to know.
Okay, thank you.
Christine, do you want to unmute and ask your question?
Hello. I was wondering, so I imagine with the simulations, it must take a lot of computing power, and I was wondering what language do you use and how much effort goes into writing the codes for all of these simulations?
Yeah, so something that I have alluded to a couple of times is choosing the right model for the particular biological problem that you're interested in. So the corollary of that is then choosing the right programming language for the particular model that you're making. So some of our models don't need a programming language at all. You solve them on a blackboard and do some equations and get some results out. For the more complex ones, so particularly the agent-based models that we were looking at, what those really are is a huge system of differential equations. So every cell has interactions with every other cell, and we describe them using essentially Newton's laws. So those huge systems are really computationally expensive. So it becomes very slow to run these simulations. And so we'd want to use something like C++ or even C to try and really get the performance of those. Whereas for a simpler model where we're not trying to solve tens of thousands of equations, but trying to solve sort of half a dozen or something, I might use something like Python that's a bit more accessible, a bit easier for me to write something quickly, but has that trade-off of performance.
Okay, thanks. John, do you want to ask your question if you unmute?
Doesn't seem to want to unmute. Oh, can you hear me?
Yes.
That's got you.
Okay. Well, I suppose the big question is, do you have an inkling yet as to how your model might be applied in helping to find a cure?
Yeah, so I think in the very long term, so I'm talking sort of decades away, I think that people will use personalized mathematical models to come up with personalized treatment. But I think we're a long way away from that yet. So at the moment, the real value of these models is in looking at specific problems and saying, can we simplify our system and ask some specific questions about how a piece of the biology that we don't quite understand works? I think where we're starting to become more successful is in doing things like modeling different treatments. So there's a lot of interest at the moment from a few different research groups about modeling radiotherapy. So at the moment, if you're prescribed radiotherapy, you'll probably get one of 2 or 3 different treatment regimes. So they'll either say, come in every day, Monday to Friday, and we'll give you the same dose, and then you can have the weekend off for good behavior, and then we'll do it again next week. Or occasionally now they're doing a more accelerated version, which is where you might get 2 doses a day that are twice as strong, but for much less of the time. But there's no real reason to think that those are optimal. And there's no reason to think that we should be giving every patient the same amount of radiation at the same time. So there's a lot of interest at the moment in saying, can we take data about a particular patient's tumor and predict what the right amount of radiation should be to give them and when we should do that? And that's very much an open question at the moment, but I think it's one that we're making a lot of interesting progress on it. And there's a few clinical trials that are going on at the moment, which are testing mathematical models in the clinic.
Okay, thank you. Thank you very much.
Thanks. And Andrew has another question.
Yep, I'm muted.
I can hear you now.
Thanks. Good. This is a slightly tongue-in-cheek question here. I loved your diagram of the comprehensive map of the mTOR signaling system. Where can I get one? It's the sort of thing I'd like to stick on my wall. And
yes,
either.
Yeah, I don't even know what the mTOR signaling network is.
that doesn't matter.
Yeah, I can send you the send you links to the paper that it's from if you'd like.
That would be great. I'd love it. Thanks.
Yeah, no problem.
By tomorrow, Andrew will explain to you what the mTOR signaling system is.
Yeah, I'd love to know.
Okay, Christine, I want to ask your question.
Yeah, collagen tumor cell interaction is probably hideously complicated. Is there ever any chance that mathematical modeling is going to help to understand this?
To understand what, sorry?
Collagen tumor cell interaction.
Yeah, so I think in terms of help to understand, it's, and I mean, this is a bit of a cop-out answer, I think. But the question is, what do we mean by that? So in terms of, making some progress towards understanding those relationships and describing those relationships, I certainly think that modeling can help do that. In terms of understanding, meaning we have a full and perfect knowledge of what's going on, I think, that's a lot further away. So it's very much this process of where you can't really get more out of a model than you put into it. So if we don't have a good biological description of what's happening and a good biological understanding, then we can't use a model to gain that. So it's not a substitute for the experiments, but it's a way of helping us to understand what the experiments do, and maybe telling us how to look at those in a new light. So as I say, that's a very cop-out answer, but I think, yeah, hopefully helps.
Thank you.
I'm going to pick up on a couple of questions that have come in from YouTube because they can't ask them themselves. So David said, thanks for the talk, it was fascinating. Are your models always in 2D? Is there any benefit to doing them in 3D?
Yeah, so they're not always in 2D at all. So although the models that I was showing you there are 2D, we've done 3D versions of them where you see very similar dynamics. The big problem with doing 3D rather than 2D, particularly with these agent-based models, is just how much more computationally expensive it becomes, because you're adding an awful lot of cells, because it's scaling with volume rather than with area, and that becomes very slow to solve. So if there's something, the real reason for doing a 2D model is because you're looking for something which is a bit more complex than you can describe with something that's one-dimensional, where you're just looking at radius, but you're not needing the dynamics of 3D in there. I do think there is a bit of a problem where it's quite common to do these 2D models and to think of things as being 2D, when actually, obviously, this is living in a 3D environment. So particularly the model which I showed you briefly, where I take the histology image, the image showing the oxygen, and use that to define a region to solve this thing on. Partly the reason we do that in 2D is because we just have a 2D image, so we don't know what's above or below. But also we do know that what's above and below has a big impact. So if there's a blood vessel that's just outside of the plane of the image, say, those cells are going to have oxygen, even though we can't see that on that image. So there is a bit of an issue there. But really, you don't want to make a model that's more complex than the data that you've got to kind of validate it. So if you only have information about a 2D slice of something, it's quite a difficult problem to say, well, can we build a 3D version where it has this as the cross section, if you like?
Okay, thanks. And Milosh has asked, do you build models to answer specific a priori questions, or do you sometimes come up with questions only after the initial EDA? Perhaps you can explain what EDA means as well.
Yeah, so I'm not sure what EDA is, so that hopefully that'll come through.
Design, maybe, I'm guessing.
That makes sense. Yeah, I think the answer to the question is, in theory, we design the model to answer a specific question. In practice, what tends to happen is we do a lot of work building on other models. So if you've got an existing model where the scientific community understands the dynamics of that model quite well, you may be better off using that as a building block to push towards whatever it is that you're trying to do, rather than starting again from scratch. Because otherwise you end up trying to replicate a lot of the same work that's already been done. So even with these tumor spheroids, there's a lot of unanswered questions where, you know, if you make the same assumptions about the tumor cells, about their proliferation rate, say, do you get the same result if you use a 1D Greenspan model versus a agent-based model written in Chaste versus an agent-based model written in PhysiCell or something else? And we don't really know at the moment. That's still work that we're trying to do as a community.
Okay, thanks. Bob, do you want to unmute and ask your question? That's Bob Clifford.
Yeah, hi. Yeah, a very interesting talk. I think you more or less answered my question, but I was just wondering, when you were talking about using radiotherapy in treating cancers, whether there'd been much work done using virtual reality as a way of looking at how things like X-rays may penetrate tissues and the relative strength that occurs in different parts of the tissue, what its absorbency, you know, how the absorbency might affect your model.
Yeah, so I think where virtual reality is really useful, or will be really useful, is that we're getting better and better at imaging these things. And so we're moving from 2D images of what that tumor looks like to 3D images. But obviously most of our visualization tools are all built for a computer screen. So, you know, you may have seen those MRI pictures where you can sort of scroll back and forward and you see someone's head come sort of in and out of focus, essentially. But really, you want to try and see all of that at once. So virtual reality is a really powerful tool for that, because you know, if you've got a 3D tumor and you can see it in virtual reality, you can zoom in on it, you can make it bigger and kind of move it around and really try and look at what exactly is going on within that. So I think it's going to be very useful as a visualization tool. And people have visualized the output of mathematical models using virtual reality as well, so that you can have your 3D spheroid growing and you can move it around and go and look at particular parts of it.
Yeah, because I know that in chemistry, it's been commonly used to be able to look at molecules, turn them and twist them in space so you can see the formations of them.
Yeah, I think we haven't quite found what it will be most useful for in kind of maths and in biology, but I think it's clear that it will be useful. And so it's a bit tricky, because we, you know, I think if you ask me this in 5-10 years time, hopefully there'll be a nice straightforward answer where we think, yeah, we all, you know, this is where it's been most useful, but I think we haven't quite found that at the moment.
Okay, and Carol, your question.
Yes, it's probably only just a brief one. Has any modelling been done on the involvement, if any, of free radicals in tumour formation?
Yeah, I don't know. So I'm not aware of any models specifically about that, but that doesn't mean that they're not out there. So I tend to be looking slightly further down the line. So I don't really research the kind of initial stages of how that tumor starts, but I'm more once it's established what is going on. But yeah, I think there are people that are researching kind of tumor genesis, and I would assume that they have free radical models, but I'm afraid I don't know of any specific ones.
No, that's fine, thank you.
Okay, and what looks like being our final question, even though it was the first one asked at the beginning of the talk, is from Steve Williams, who asks, which Chelsea defender should I target?
Oh, none of them. Get the attackers. Yeah. Yeah, if anyone's particularly interested in fantasy football, I'm doing a talk on that tomorrow night.
That was something I mentioned in the introduction. Some people might have missed. But you might like to explain what that was about, Josh.
Oh, yes, so there's a fantasy football competition that runs every year, and I won it this year out of 7.5 million people. And the next season starts on Saturday. And so suddenly I keep getting asked to do all of these talks about fantasy football and talk to people about Chelsea defenders. So it's been quite nice talking about my actual research, to be honest. It's a pleasant change. It'll all go back to normal next week.
Okay, well, that brings us to the end of the formal meeting. Many thanks to Josh for a fascinating and enlightening talk, and to you all for attending. A reminder that if you wish, you can donate to paypal.me/WinCafeSci, and that might enable us to fund our next promotional campaign.